Okay, so this is a figure from a recent paper from the Trapnell and Sandura labs, where they combined a bunch of data sets over a few million cells to essentially map out the trajectory, all of the trajectories of mouse embryogenesis. And the point I'm making is that single cell data has led to these advances in being able to map cell dynamics at an unprecedented resolution. And so the next natural question is, what's driving this? What are the mechanisms behind this? And there, one of the key insights is that because of the heterogeneity of single cell data, you can get to causal mechanisms just from observational data. And these will be hypotheses that you have to test, but these are fairly high quality hypotheses. So let me give you an intuition. So in this cartoon, the blue stem cell is differentiating into two cell types, the orange and the pink. And the intuition is that if you have a tissue sample and you assay it, you'll pick up cells at different stages. And because of that, you should be able to get the gene regulatory network that's driving this. And of course, ideally, you want the network in its full detail. So for example, if there are combinatorial effects, like, for example, here, G3, oops, sorry. Here, I'm not going to point because this mic will go away. So here, G3 has a combinatorial effect coming in from TF2 and TF3. So you want to pick up things like that. So let's take a look at how these kind of analyses are set up. So once you have your tissue sample and you do your sequencing, what you get is a bunch of numbers, just raw readouts. So the first step you have to do is to go from these numbers to some measure of dynamics. You have to figure out what the underlying, dynamics of the cells is. And you can do that either through trajectory estimation in pseudotime, or if you happen to have spliced and unspliced transcripts, you can do that through RNA velocity. And once you have this dynamics, then the next step is you infer causation. You go, okay, what are the underlying regulatory mechanisms by which this dynamics was generated? And the problem I'm talking about is that these two steps are not in sync. So what do I mean by that? So if you look at the methods that currently exist for inferring this causal connection, and there's a whole host of them, they are very diverse in some ways. There are methods that look at random forest, there are methods that look at regression, methods that look at, for example, a dynamical differential equation type setup. But in one way, they're all very similar, which is that they all trace their roots to time series and dynamical systems analysis. And they all have this assumption that the observations you have, they can be lined up in some time axis on which you then perform your causal analysis. And that is a problematic assumption, because that is not supported by the previous step of inferring dynamics. So let's say you had your data, right? Going back to my cartoon example. And you did a perfect estimation of dynamics. And so you got the trajectories exactly right. Your cells are placed exactly right. What these methods will end up doing is they'll take all of these cells and squish them into one line. So they're correct in saying that, for example, the orange cells are downstream of the blue ones, or that the pink cells are downstream of the blue ones. But they also end up saying these unsupported things about orange cells versus pink cells. And that leads to incorrect inferences. And Velorama is an approach to essentially address that. And by the way, it's not just about branching trajectories. Even if you have just one trajectory with no branches, one issue is that pseudotime is not time. So 1 unit of pseudotime at the beginning of a trajectory is not the same thing as 1 unit of pseudotime at the end of a trajectory. And many of these methods treat those as fungible, and that's not valid. And of course, the big issue here is that when you go to RNA velocity data, this assumption of a single time axis just does not work. So with RNA velocity data, you essentially are getting these individual measurements per cell of spliced and unspliced transcripts, which give you a local direction of where the cell is going. And you can combine that with other analyses. So for example, there is this method cell rank from the Fabian Thies lab, which can give you a cell-to-cell transition matrix, saying, here's where one cell is going, and these are the cells it's likely to go into. That kind of dynamics is just not compatible with some kind of a linear time ordering. So here's our fundamental insight. What we are saying is we shouldn't think about differentiation as a global phenomenon. We shouldn't be trying to place one cell against all other cells in the data set. The dynamics models essentially are giving you information about one cell against some cells in its neighborhood. And you need to be able to work with those local informations. And the way we do that is by going away from total orderings, which are very restrictive, to thinking in terms of partial orderings. So in particular, instead of trying to model the dynamics as a single linear time axis, we think of a directed acyclic graph. So in this graph, an edge is going from a cell A to a cell B if A and B are similar, and the direction of differentiation from A goes towards B. And so if you look at this DAG is also saying that orange cells are downstream of blue ones. It is saying that the pink cells are downstream of blue ones. But it is not making any statements about orange versus pink cells, which is what we want to do. So of course, the question then is, once you have a DAG, how do you do inference on this? And we go to this idea of Granger causality. So Granger causality is a statistical concept, a classical statistical concept, which tries to infer some notion of causality from observational time series data. And so the idea is, in a dynamical process, a cause precedes effects. So conversely, if you can identify predictive lead-lag relationships, they have some possibly causal explanation. And this might not always be true, that's why it's a hypothesis, but over the decades of usage, Granger causality has proven to be quite useful. And even in a single-cell gene regulatory context, there are methods like Sincerity and Sinch, which use GRN, but they do run into this issue where classical Granger causality requires a single time axis on which you can do this analysis. And so one of the key conceptual contributions we're making is the idea that let's generalize the notion of Granger causality from total orderings to partial orderings. So classic Granger causality is formulated like this. You have some dynamical system, it's a multivariate dynamical system, so you have, say, X and Y. And you say X Granger causes Y if, to predict Y's value, it is significantly useful to be able to have access to X's history in addition to Y's own history. And that significantly is where you put in a bunch of math, you can do nonlinear things, whatever you want to. But the idea is you say if X's history is meaningful to Y, then X Granger causes Y. In a DAG context, that's a very natural generalization. What you see is, okay, I have X and Y at this node V, and if Y's value at V is improved by making use of X's value at the ancestors of V, then we say that X is Granger causing Y in this DAG context. And note that if you were to go to linear chains, this basically collapses down to the classic definition of Granger causality. And it has this odd notion of lookbacks or lags. So you can look back 1 hop, 2 hop, 3 hops to see how much far back in history you want to consider X. Okay, so this is what Velorama does. You start with some single-cell observations. You do either pseudotime or RNA velocity. You get a DAG, and then we set up a neural network to do inference on that. And I'm going to show you some results, but I want to give you some sense of what things look like and why RNA velocity is powerful. So this is synthetic data from Sergio. And here, each dot essentially indicates that regulator is interacting with that target. And so from the synthetic data, you can simulate transcript counts, you can do pseudotime analysis, and this is Velorama on that data, and it does quite well. I'll show you actual numbers later. But if you were to instead simulate RNA velocity data, splice and unspliced transcript counts, it does substantially better. And in fact, when you do these transition matrices from RNA velocity, those can be probabilistic instead of binarized. And turns out Velorama can take use of that as well and get an extra bit of juice. But the overall point I'm making here is more that RNA velocity is powerful, and Velorama can sort of help you make use of that. So I'm going to briefly talk about how we set up the neural network here, right? So essentially, you first set up the DAG. And for any gene, essentially, we are setting up a neural network where we are trying to predict gene G's expression as a function of its history at 1, 2, 3, how many lags you define, and all transcription factors at that lag. And then you set up a regularized framework where the result of this analysis is transcription factors and lags. So you don't predict just transcription factors, you predict TF, lag pairs, which are predictive of this gene's expression. And just to go into this in a little bit more detail, so the first question is, how do you come up with the DAG in this setting? So if all you have is pseudotime data, then the way we compute the DAG is you first set up the k-nearest neighbor graph. Then you use pseudotime to sort of essentially say, hey, let's order the edges to be consistent with that. So you end up getting a DAG. If you're using RNA velocity, you can use the transition matrix. There is a bit of a catch where there can be cycles. Turns out it's not that big an issue. It's in the paper, and you can deal with that. And then one thing that we do here is that we pre-compute the contributions from all the lags and store that as input features, which is slightly different from some other work we have done where we do this in a graph neural network setting, and there are reasons for that, and I can talk about that offline. But the next interesting point is the way we set this neural network up, the first layer is special. That's where we put in that idea that we want to find the input features that are significantly predictive. And we do that by introducing this regularization. It requires a little bit of care in terms of optimization and so on and so forth. But the bottom line is you essentially get, at the end of this, transcription factors and lags, which are significantly predictive of this one gene. OK, so let me show you some benchmarking numbers first. So this is all based on synthetic data where you do have ground truth, so you can do an apples-to-apples comparison. And this is like on a standardized Sergio synthetic data set. And I'm first going to show you Velorama in the context of pseudotime data. And Velorama here is the olive bar on the far right, and just next to it is Genie. And as you can see, on an AURC basis, Velorama does better than many of these methods. But what's even more striking is if you look at precision and recall, the outperformance is even more stark. And in this context, AUPRC is the right metric because the true positive, true negatives are massively unbalanced in a gene regulatory setup. And in fact, if you were to now say, instead of using pseudotime, what happens if I use RNA velocity? You end up getting an even bigger boost. So we ended up changing the axes here because the performance changes. So so now on here, you are seeing 3 columns of Velorama. The light blue that is covered in red over there is basically the same thing from the previous slide. And the 2 darker blues are essentially Velorama with RNA velocity. And you can see how substantial a boost RNA velocity can get you. And again, if you use the probabilistic transition matrix, that ends up getting you a bit more of a boost. OK, so this sort of, and the other interesting thing here, just as a note, is that the more heterogeneous the data set, the stronger the outperformance seems to be, which was interesting. So now I'm going to spend a few minutes talking about this idea of lag. So there are many things you can do with these methods. One thing that we went down a bit of a rabbit hole into is looking at what is the impact of transcriptional lag. And so one simple analysis we did was look at some well-known RNA velocity data sets. What we found is that transcription factors involved in differentiation seem to act more slowly than others. And we thought this might have to do with the theory of pioneer transcription factors, which pull in other transcription factors, and so they might take some time to sort of show their effect. And we got a list of pioneer transcription factors, and nicely for us, that seemed to show up, that pioneer factors are actually slow. So then we went in and did a deeper analysis of human corticogenesis. And we looked at a data set where we also had multimodal attack-seq data, so that we were able to validate some of what we were saying from that chromatin accessibility perspective. So here, we looked at this data set, we performed our analysis, identified transcription factors that are fast and slow. Turns out the lags that we estimate are consistent with the lags that you could estimate using, say, chromatin accessibility versus gene expression. So that gives us some comfort. But I want to spend a few minutes just talking about what we found, because I think it's really cool. So we took the 10 slowest transcription factors and the 10 fastest ones. So what I'm showing you here is that the fast transcription factors seem to be differentially expressed in brain tissue, and this is based on ENCODE type data. That is not so much the case for slow transcription factors. And actually, you can make a stronger statement. Fast transcription factors are not only differentially expressed in brain tissue, they are actually cell type specific within brain. So if you look at the motifs for these transcription factors, they seem to be specialized to brain tissue types. Not so much the case for slow ones. And then, because Velorama can give you combinatorial answers about transcription factors, you can see, well, what kind of cofactors are being involved. And sort of what you would expect from like a pioneer transcription factor type theory, it turns out that slow transcription factors seem to disproportionately like to work with fast ones. You don't see this much for fast transcription factors. Okay, so you do this. Now, what is the impact of this on disease? And what we found is that fast transcription factors are disproportionately more implicated in neuropsychiatric disorders, while slow ones are disproportionately more implicated in cancers and gliomas. So like one story here is the slow ones are chromatin remodelers that are not necessarily tissue specific, but they start the whole thing going. And if they go wrong, you have cancer. The fast ones are tissue specific, cell type specific. If they go wrong, you have cell type specific things. And there's a few more things I can talk about, but I'm going to cut short. If there was one takeaway I could offer from this is I think we should model dynamics as a local thing, not as a global thing. I think Granger causality and a partial ordering can offer you flexibility and power. And thinking about the lag of transcriptional regulation, I think, is a deeply functionally relevant thing that we should be looking at more. I'll skip this. I want to thank my co-authors, Alex, Anish. Anish was a high school student, now starting at MIT, and Bonnie. And I'll be starting a new position, so if folks are interested in working with me, please come see me. Thank you.